So next, we're very lucky to have Dr. Tim Bray. I worked with Tim at Cobthorne with Sheppy for a number of years. Tim's a lecturer in conservation science at Bristol Zoo. He's a conservation geneticist specialising in using molecular genetics approaches to answering questions in conservation, ecology and the distribution of biological diversity. And his talk today is Farm to Forest, Conservation Genetics of Domestic Animals and Captive Population.
Thank you very much. Brilliant. So really this whole talk is just an excuse to show off my newly acquired Burmese bantams, which were from Sheppy, and it was one of his passions, and I couldn't imagine them going anywhere else or anything else happening with them. So we have taken these over, and they're now causing havoc to our neighbours. So what I want to talk about, the reason I've called it From Farm to Forest is because Sheppy used to give a talk on From Forest to Farm, so where he talked about domestication and he related it all to the Cobthorne Trust and sort of the aims of the Cobthorne Trust, which was all to do with the research of the domestic animals and their wild relatives, how you actually use their particular traits, integrating them into kind of the greater environment, actually, you know, things like conservation grazing. And then obviously, I don't really even need to say this, but Sheppy was all about promoting the use of different breeds, environmental awareness. He was always running off to DEFRA to talk to them. He was always very frustrated that things are happening and changes in Europe, and you know, his voice isn't being heard strongly enough. So this is really very much inspired by Sheppy's work, and I think it's really appropriate that I'm after Rex, because one of Sheppy's whole presentations was all about sort of the changing selection pressures by humans in actually, you know, looking at the morphology of these different breeds, and obviously there are lots of them, and please don't quote me on numbers, but there's lots of different time periods over which we've been domesticating animals. So this is very much a bit of an overview, and I'm going to go into some painful detail about some population genetics, but please bear with me. So really, what we did is we took wild ancestors, you know, things like red junglefowl and mouflon, and then we found these characteristics that we wanted, and we started to breed lots and lots of different forms. And then once we'd got different forms, we started to kind of imagine there was a pure form, and we've already had mention of breed standards and ideals and this sort of thing. And this is very much a process of looking for characteristics in organisms that we want to utilize or promote and then perpetuate. And it's not just about the organisms themselves. Actually, it's about how they fit into our farming landscape. I had to find a place to put these in. Sheppy was really, one of his great passions was getting the cattle up onto Cadbury Hill and getting them to graze the nature reserve, and the stories about how when the Dexters were left to themselves, if there were young ones, they would form a little protective ring with everyone facing outwards and the young ones in the middle. They would graze, their own management regime, taking some of the brush away, you know, cropping very patchily, creating habitat for invertebrates, you know, mammals alike. And the same sort of thing at the sea cliffs. You could just leave the soways to do what they did. They're not going to go and throw themselves off a cliff when there are dogs around. They'll just sort of run to an inaccessible area. So it's all about kind of this synergy with the landscape, and it doesn't have to be, agriculture doesn't have to be seen as being this completely detached entity from conservation and from the environment. So now I want to talk a little bit about genetics, just insofar as what we've been doing is we've been looking at the results of the genes when we're actually selecting for particular traits. And you can actually, there's lots of contemporary studies where they're looking at different organisms and they're saying, well, what are the underlying properties, you know, what are the underlying genes that are controlling what we're seeing in our domesticated animals versus the wild? And this is a great example, I think, which is the grey wolf And what they've shown is that the process of domestication actually can be seen on the genome of these guys in a particularly large effect gene. This idea that wolves, wild wolves, they have a copy of genes for salivary amylase, so sort of digesting starch, but they don't use it so much, so it's not like they've got lots of copies. But if you look, so they've got sort of a single copy, it's this blue bar in the top corner. And then if you see the domestic dog has loads and loads of copies, because actually one of the things that domestic dogs did when they kind of started to associate with humans was take advantage of human food sources. And I've heard ridiculous stories of cats eating tomatoes in rubbish dumps. But, I mean, dogs would have been making use of starches, and there would have been an advantage to having more copies of a gene producing starch digesting enzymes, as well as the regulation. So down at the bottom, it's about gene regulation. So we're not just talking about the presence of genes, we're talking about how the genes are working. So what's great is that we can actually see the signature in the genome of domestication. And we can start to say, well, let's actually look at what's going on sort of underneath the skin. And, you know, it's something that everyday breeder won't be wanting to do this, but it's something that we can actually investigate now. So along with the positives, there were also negatives. I apologize for showing a really horrible slide, but this is something that can be really, really important. You get single genes that can have negative effects. So these are things we can kind of avoid. And actually, if you can pinpoint things like this, you can really improve your breeding program. And this is something that Sheppy would do on the Copthorne farm. You know, you identify individuals that share, that have a gene that potentially, if put in combination with another mating, can produce such a bulldog fetus, which is sort of a 1 in 4 chance if you get the 2 carriers. Then you just breed so that you can avoid this sort of thing. So you can still maintain a selective regime, you can still have your population, you just know what to look for. And actually the genetic testing in this case is really, really important. But what's funny is you also see behavioral responses. It's not just, you know, physical traits, it's not just sort of coat colors and things like that. You can actually see changes in behavior. And I had to find a way of putting this in because it's a brilliant example of conservationists with a small population of a wild creature saying, well, let's bring this population back, let's do some manipulation, let's captive breed it, and try and get this population to expand in size, you know, to its previous levels. But what happened here was, because of the small population, we've already heard the term fixation being thrown around. So there was close to fixation was this allele that meant that the birds didn't really care where the egg was around the nest. So they're laying an egg, and they're kind of sitting on the nest, but the egg could be slightly over to one side and not getting incubated properly. And this is a real problem. So this actually reduced the survivorship, this particular bird. And here we have an example of an egg that's kind of over to one side. And that's really important. So there's this whole conservation program, and just over to one side, just because there's a small population, and the impetus is on getting numbers up and not particularly selecting for traits. There's been a relaxation because every time an egg rolled to one side, there'd be a human there to put it back. So it's a very strange situation, but then you get the wild population with a very low recruitment, and you have to think about, well, you know, there must be something we can do about it, and you know, identifying what's going on is one way of doing that. So it's almost like I'm saying that everything you see is affected by these large effect genes that you can identify on the genome. But that's not really true. And that's why, in some respects, sort of turning a blind eye to the actual fine details makes life a bit easier, because you get, there are classic pictures of sort of size distributions with these quantitative traits where you have lots and lots of different parts of your genome which are affecting a single condition, like your height. And that's probably the major condition. That's probably the majority of traits you find will be determined by many different loci, many different genes on different parts of the genome. So we have to really consider this because we're dealing with very small populations. And I've got this perhaps horrible graphic, but it's just to give you an impression that if you've got a certain number of individuals back in time, then if it's a small population, over time you might get these drastic changes in gene representation in your population, and even if you do have a process of selecting only a single male to carry on from a litter, that sort of thing, even if you're very careful, with very small populations, you can have problems with these random fluctuations in your genetic composition. And it's not completely independent of your selection regime, but it can be largely independent of your selection regime. So what I just want to talk about here briefly is something we did with the dexters, but it's also the process of how you kind of move away from looking at phenotypes and you try and represent as much of the genome with looking at the genetic composition itself. And in doing that, you can use as much or as little of the genome as possible. So obviously, it's much simpler, and I've been left behind somewhere about here, and genetics has carried on without me, I feel, somewhat. But back in the day, when I did my PhD, we were looking at sort of 10s or 20s of loci, and now we're looking at millions. So what happens is we're getting a really good representation of the genome, and we can actually start to sort of home in a little bit on even these tiny little contributing genes that are doing things that we see the phenotypic effects of. So this shows you that we can actually start to take anonymous or neutral markers to get some idea of what our populations are doing without necessarily linking them directly to particular traits or anything like that. So it's kind of an approach of looking at the demographics of a population. And that can be really useful and really interesting because we have populations which sometimes they're being bred for very different things. And we've already heard that said by two people. People do breed. Breeders breed for different characteristics. And the dexter is a great example, I find, because the dexter has all these different types. It has the leg length that we're talking about. So they have short legs, the sort of in-between medium legs, long legs. You have different colors, which different breeders will sort of turn their eyes away from and be a bit offended by. You have the animals, the polled animals, you have the horned animals. So there's all these different conditions, and the different breeders are breeding for completely differently, which makes it really, really interesting. So what we wanted to do is just take a genetic model and ask some simple questions about, so this is just a population model. If you imagine you start off with an ancestral population, it splits into a couple of breeds, and then you can see, well, what happens if those breeds come together? Can we start to look at our breed composition today and ask the question, well, can we prove, whether there's a pedigree or not? Can we prove what was involved in the creation of the breed structure as we see it today? Because actually that's kind of interesting because we can talk about how that relates to other populations, and if we could model it really, really nicely, we could just take some DNA from a population and say, well, we might be able to see exactly its population history without having to go there and having to sort of go through the history books. So there's lots of different ancestral populations. I mean, breed diversity is absolutely crazy. And the really interesting thing about populations such as the dexter is that they have really nice, like domestic animals are brilliant for their recorded history. So we can really use these as kind of this springboard to ask questions you can't ask of wild organisms. And potentially you can sort of push those, the information you get into the management of wild populations as well. So what's great about the dexter is we kind of have some idea that it came from a couple of source breeds, or they were major contributors. So we can start to model the dynamics going on, and we've also got ideas of the population changes. So we've got size population. If you imagine the blue is kind of some representation of the population size of the breed. And then we have these times during which populations were maybe very small, different populations were involved in contributing to the breed. So we can start to ask questions about, can you identify if you've got, say, we've talked a lot about pedigrees this morning, if you've got a pedigree inconsistency, or if there's another population involved and you want to perhaps quantify that. And this also works with wild populations. You can start to ask those questions about wild populations as well. And the answer is very complicated. So actually, you think, oh, it'll be great, there'll be this simple answer. And it's never like that, of course. So what we have here is we've talked about this, this random mating and this sort of breeding population. So if you look at this, then each of these upright bars is a single individual, and each of the colours represents what the computer program has decided without previous information. It's decided that's a population. And what a population means is really just a group of breeding individuals. And yeah, obviously understand that these individuals are all geographically spread around the whole United Kingdom. And we're trying to work out how you can separate this data set up to ask questions about sort of population history. You can look at founding bloodlines. You can look at how does it compare introgression. If we could separate out groups, subsets of this population, can we say, well, this has clearly had an influence from the Kerry breed, or the Devon breed, or, you know, the American population is a little bit different because it's had a differential input from different bloodlines. But actually, it's really, really complicated. And as you might expect, you get this sort of real mishmash of populations and breedings and mixings. So we kind of hit a bit of a wall with the Dexter because it's such a complicated situation. So what I'd rather do is talk about the Lincoln Red breed, just because it's a breed which actually has a couple of nice admixture events with reasons. So particularly an admixture event to sort of beef up the breed a little bit, and an admixture event to pole the breed. So we've got a few herds, and we don't have problems with massively fluctuating population sizes. We've still got small population size, which is a big problem because you get things like changing allele frequencies out of your control, you get loss of diversity, you get fixation of alleles that, you know, sort of almost willy-nilly. But you get a much nicer result. And it may look to you just like still a very messy result, but you can actually say, you can actually start to sort of partition your populations with respect to the inputs from the different ancestors. And that can be really nice. And actually, although it still looks a bit messy, you can start, when you're doing this modeling, you can actually start to quantify it. So you can quantify and you can compare to the pedigree. And this means you can actually, once you've decided your model is functioning okay, you can start to test other pedigrees. So to start off with, you have to be fairly sure of your pedigree. But it's nice in the way that it's not dependent on the pedigree in any way. So you can see actually some of the numbers you get, if you compare them to the pedigree, they're really, really close. So this is, for me, a bit of a surprise because you're going back in time a number of generations and you're asking, you know, with the DNA we've got now, can we look and see what the involvement of different breeds are in the history of this one population? And the answer is kind of yes, to some extent, and it's a really promising result. It's kind of interesting in the fact that you can take this and transpose it into other situations, and you can say, well, what are the influences of a particular selection regime? And you can look at lots of things. So one thing I didn't point out before was, I'm just going to spin backwards slightly, is to show that there are particular bloodlines you find in the Dexter that separate out quite quickly as isolated populations because they've been under their own selection regime. So what you can see is these sort of line-bred separated populations have been worked on separately, and you can actually see the genetic signature. So once you've done this once, you can take applications like this and you can say, well, we suspect that's happening in this case, so let's just do a comparison. And that's kind of a really nice result from that. So how can you use that information? So you can use it for management. You can use it to compare between different populations and say, well, in this situation, our model suggests that perhaps these populations are separating out too rapidly. There's not a mixing of the whole breed as an entity, as a contemporary entity. So let's perhaps make some management advice. Let's sort of move animals around a bit more. And this can be applied whether you're talking about sort of zoo populations or domestic breeds. And you can inform things like best practice. There are lots of guidelines for everything. And we've already talked about how things like mating systems are important. So whenever anyone mentions artificial insemination, that makes me think mating systems, just because it's changing the number of individuals that have an input into the next generation. So the potential of AI to completely revolutionize in one generation a breed is quite strong. And I equate that completely to, I've got some pictures of mole rats, because they're really, really interesting in the fact that the naked mole rat, the sort of saber-toothed sausage, as it's called, is incredible. It's eusocial, so it's like sort of a bee or something. It's got a queen, and she will be producing all of the offspring for the whole colony. And that means your respective inputs into the next generation, your effective population size, are very different to something like the horrible Cape Dune mole rat, which is this aggressive, nasty, promiscuous mammal that lives in its burrow and fights off anything apart from, a couple of seconds of mating, and then when the young are big enough, it chases them off too. So, these are completely different systems. And I equate that exactly to actually how we're managing captive populations. And I think looking at sort of population modeling in this respect gives us a little bit of information, albeit a bit of work to get to the point where you might argue that, the breeders they're doing this naturally without any idea of what the DNA is doing. So now I just want to go back to the ancestors a little bit and talk about, well, what you could do with these methods is, because you're going back in time, you can actually use it to perhaps look at influences over landscapes. You can take just a few samples and you can start to model what's going on in regions, if there are populations expanding to new areas, or if there are very different populations that are mixing because they've been, for example, involved in the pet trade. And you can even start to identify where these individuals are coming from and potentially to take them back there. So what's really amazing is you get situations like monitor lizards in Southeast Asia, and you find that every single island has its own unique sort of haplogroup, if not unique species or subspecies. So you've got really sort of strong genetic diversification, and when you're using a neutral markers modeling approach, then you can start to say, well, this isn't exactly the same as that, but if we do a bit of an admixture model, it looks like a mix between them. And that brings me to kind of the freshest part of this that's in my mind is what this sort of genetic modeling approaches is doing really right now. The World Pheasant Association has been doing some incredible work with genotyping species, because they're looking at captive populations, they're looking at how do we breed up populations, how do we manage them, what is the best way to include individuals that potentially don't have a pedigree? Because if you don't have pedigree information, how do you reclaim individuals? And this was a big problem that Andrew was talking about with, people don't register their animals for 1 year, and suddenly that, maybe you need to have an amnesty and say, right, come on, we need to get the breed bigger, come on, let's bring in some animals from elsewhere. And you can get these, you can start to sequence large amounts of the genome, and this is just what the World Pheasant Association is doing at the moment, in trying to increase the populations of really, really endangered species, and to try, we've got known individuals that are pure, but you need to know that somewhere out there, those individuals that you don't know any information about, are they still able to be sort of retracted back in and used in a breeding program? And I think you can see us quite nicely in this case here. These data are absolutely brand new. And you see these little, on this plot here, it's just an idea of just some geography of the data, and you've got these little black crosses, which are these individuals we don't know the provenance of. And we're asking, are these individuals a member of this species or this species? Or are they slap bang in the middle, which means we probably shouldn't be using them because they're probably a hybrid, even if they don't look phenotypically hybridized. And it looks good. So actually, this probably is expanding our population, and we can start breeding with them. So that's kind of a utility. But to go a bit sort of full circle and wrap it up, I would like to say just that actually it makes sense to start to think about these approaches, to start to think about how, you know, the next generation of selection can be considered. And it could be to do with anything. It could be to do with disease resistance. It could be to do with, you know, trying to account for changing climate. And I don't know how that would manifest itself. Potentially, if you're looking at a climate getting wetter, you want to, you know, avoid feet getting rotten and that sort of thing. There must be sort of traits related to that. So you want to know if there's a genetic basis for a particular condition that you're looking at in your organism. If you can find some large effect genes that you can actually start working on, and maybe groups of genes that you can work on together as a block, so you can sort of maximize your effort and be most efficient. So if you see in this particular plot, it just shows the entire genome, and there are all these sort of lines represent genes, potentially genes that you could identify and start selecting for in your breeding program. And these ones that have risen really high, they're ones that there's really a big difference between individuals or populations, suggesting that it's important. You know, if it's really changing, that's probably natural selection taking hold and carrying that variation forward because it's got some utility. So, you know, potentially this is something that we're looking at. That's all I want to say. Thank you very much. I want to acknowledge these and also thank Philippe for organising this so nicely. And just say, yeah, are there any questions? I don't know if we've really got time.
Yeah, well, thank you.